Okay, so I think we can start. So welcome everyone to today's edition of Tissue Talks. It is my real pleasure to introduce today's speaker. It is Jeff Hubbell, who is really a giant in the area of immune engineering. He started his career at Kansas State University, where he got a bachelor's degree in chemical engineering and went on to get a PhD in chemical engineering at Rice University, working with Larry McIntyre on a project that was focused on thrombosis. He started a faculty position at the University of Texas in Austin right after receiving his PhD without a postdoc, which is an unusual, pretty rare situation. A few years later, he moved to the Caltech. This was in 1995. And then 2 years later, he was recruited to the Swiss Federal Institute of Technology in Zurich and to the University of Zurich. In 2003, he joined the Swiss Federal Institute of Technology in Lausanne, EPFL, where he served as the founding director of the Institute of Bioengineering and also as the dean of the School of Life Sciences. In 2014, Jeff was recruited to the University of Chicago to their Pritzker School of Molecular Engineering, where he was very much involved in building the immune engineering program. So we are talking about 2014 year. Very recently, last summer, he was recruited to the New York University Tandon School of Engineering to serve multiple roles. He is vice president for developing bioengineering strategy for the for the University of New York, NYU. He is the lead of the Initiative to Advanced Engineering and Health, and he is professor of chemical, biomolecular engineering, biology, and also medicine in their medical school. This cross-institutional initiative that he is leading altogether is expected to develop a strong focus in immune engineering in the context of multiple conditions from autoimmunity to inflammation, allergy, transplantation, neurodegeneration, cardiometabolic diseases, tissue repair, etc. So he will speak today really about two of this area that are related to immune engineering. Jeff's work is highly impactful that you can clearly see from his over 90,000 citations and a very remarkable impact factor of 159. If you look into papers that he was publishing a couple decades ago when some of these areas that I mentioned were still evolving, you will see two classics. One of them was about synthetic biomaterials used as instructive extracellular microenvironment for morphogenesis in tissue engineering. This is a paper that he co-authored with Matthias Lutolf and published in Nature Biotechnology. And the other one is about synthetic matrix metal protease sensitive hydrogels for the conduction of tissue regeneration. In recent years, he developed two very important approaches. One is the approach to inverse vaccination for treatment of autoimmunity, and the other one is the immunotherapy delivery system that works in a way that the therapeutics finds the tumor by seeking out and binding to the tumor collagen. This is really a very unique approach. He was very extensively recognized and awarded for his accomplishments and his leadership. And I will just mention that he is one of the very rare members of all three national academies, engineering, medicine, sciences, and he's also a member of the American Academy of Arts and Sciences. Jeff, thank you so much for being with us, and you can now take over the control of the screen.
That's great, Gordana. Thank you so much for the opportunity to, let's see, you have to stop share. Thank you for the opportunity to join Tissue Talks, and thanks also for just running this series, which has been is really really so wonderful for so many scientists. See if I managed to share the right screen. You see
Yeah,
full screen now?
yes.
Great. Yeah, so really a great pleasure to be able to join. So as Gordana said, my lab, we think about immunoengineering, and immunoengineering relates to regeneration, relates to autoimmunity, relates to immuno-oncology, relates to vaccination, sort of a number of areas. Given that this is tissue talks, I'm going to try to focus in on more tissue things. So I'm not going to talk about vaccination. And I also, although near and dear to my heart, this concept of inverse vaccination, meaning vaccine antigen-specific therapies to induce regulatory T cell responses, which is now in a couple of clinical trials. I'm not going to talk about that, but I'll focus more on tissue-related things. I'd like to start out actually with some of our thinking from a land far away in a time long ago, that still relates to what I'm going to talk about. So Gordana mentioned, some work that Matthias Lutoff did in my lab many years ago. I'm not sure if he did it with me or if I did it with him, but fantastic scientist. But we were thinking about contexts of signaling. A context of signaling. I'm going to go to more modern stuff in a minute. Don't worry. Context of signaling. There were physical, meaning immobilized signals, soluble signals, and maybe soluble, maybe not soluble signals, and then asking how the context of all this relates to signaling. We were in particular interested in growth factors. There was a lot of work at this time in growth factor therapeutics, trying to develop growth factor therapeutics that wasn't really all that successful when it came down to clinical trials. And we were wondering why not. And many people were studying growth factor biology and cytokine biology by sprinkling the molecules in media, or rather like putting salt in my soup. And we thought, well, gosh, that's not actually the way these molecules have evolved to operate. Rather, they operate by being immobilized transiently, reversibly, most of the time anyway, reversibly in the extracellular matrix. And how does that change their signaling? And then maybe how might that change their translation? So I want to set up many of my thoughts by going back to these older ideas, which still haunt my mind, occupy my mind. So how does the matrix interact in some dynamic way with cytokines in signaling to control the signaling outcome? And so I just want to like fly through some really old work that I still think is very interesting. This was done by Mikhail Martino and Priscilla Briquez, and Mikhail's now in Monash, and Priscilla is at the University of Freiburg in Germany. But we were wondering about how these sorts of interactions occur between a cytokine like a growth factor and the extracellular matrix. Actually, back in those days, most of it was thought to be mediated by interactions with the heparan sulfate proteoglycans. Sorry, just the wrong button. But we looked at protein-protein interactions, and this was the old days. So just by hard ELISAs, setting up a library of extracellular matrix molecules and a library of growth factors, and then looking at binders of A to A, B, and C, and so forth. Skipping all the details, which you can read if you care to, we identified a binder that was quite high affinity, not the only one, but quite high affinity from placenta growth factor 2. It was called a heparin binding domain, but it turned out that it bound in a heparin independent manner many extracellular matrix proteins. Collagen is its own class of extracellular matrix protein in this sense. And it was particularly convenient for us in protein engineering in that PLGF exists in two splice variants, PLGF1 and PLGF2. And PLGF2 has an insert in it that's cationic, this domain illustrated in red. which turned out to bind strongly to many of these extracellular matrix molecules. And if you scramble the sequence, all that binding went away. So that would allow us then, and this is only 21 amino acids, so it would allow us to take growth factors that bound weakly to the ECM, like VEGF-A, for example, and then to swap out the weak binder with our strong binder from PLGF2. Or when we didn't know where the binder was, we could, or taking a total non-binder, we could just stick it on the end. And we'd have to think a bit about how to do the details there. So we looked in that day, as I said a long time ago, at VEGF, which had failed clinical trials in wound healing, and also in myocardial revascularization because it caused vascular permeability. At PDGF, which was used and still is in Regranix, a drug, but for wound healing, not terribly effective. I'll show some results with that actually in murine models, and then also with BMP2, which is used successfully in spinal fusion and other bone repair indications, but in mega doses, doses that physiologically don't make sense. And so by making these fusion proteins, we could take, for example, a weak binder, VEGF binding to fibronectin at 97 nanomolar KD, and take it back. So those are all around 100 nanomolar KD, right? We could take it down to like 10 nanomolar KD, which is about where PLGF2 itself binds. So you could confer a high binding affinity to low binding affinity. Just as an aside, many of these growth factors exist in splice variants that do or do not bind matrix. And the same thing with cytokines, CCL19, CSL21, that do or do not, similar do not bind matrix. So, but just to get to the point, we studied signaling quite a lot, and I'm going to come to that the details in a moment. But one could see very big differences in outcomes. This has been a DB mouse model of chronic wound healing, or rather in the mouse delayed wound healing, and so you can see it at the same molar concentrations of engineered growth factors versus wild type growth factors, with no growth factors at all. And the residual wound, the wound went from black to black, and the residual wound goes from black to red and black to red. So this part in the middle is not re-epithelialized at the sac point. So you can see really big differences, and of course that was supported statistically, and we did nice microscopy and could see differences in angiogenic response. And this was done with Melanie Schwartz's lab. If you look at one of the Achilles heels of VEGF as a therapeutic was its induction of vascular permeability when it was given to patients in the context of by infusion in the coronaries, but in the context of myocardial revascularization, it caused a pulmonary edema due to creating vascular permeability. But here you can look at the vascular permeability that's induced with dextran leaking out of the vessels. This is in the ear, skin that's pulled back. With the wild type growth factor, a lot of vascular permeability, but with the matrix binding one, not so. So one is able to correct some of that. So this motivated us to think about why this happened. At the beginning, I didn't really know, but I want to come to the point that drives us, which is tonic signaling. That if you think of the signaling dynamics of a receptor, sorry, whether it's a receptor tyrosine kinase, like in the case of these growth factors, or another receptor, that's, for example, with immunological cytokines, the endocytosis step is more often than not the off signal. And so how could we get a prolonged on signal? We could either tether the ligand to the matrix, as I just showed you with the PLGF2 domain, or we could even tether the ligand to the cell surface of the cell. So we could tether it either way. The idea is one way or another, how do we delay endocytosis of the ligated receptor, leading to destruction of the cytokine and recycling of the receptor? So this case was done, started actually back in the Lausanne days and finished at Monash by Mayumi Mochizuki and Mikhail Martino. So where in this case, rather than anchor a growth factor to the matrix, we anchored the growth factor to the cell surface through a non-signaling receptor, here syndecan. So laminin, a big complicated adhesion molecule, of course, matrix molecule turns out to have a fairly high affinity syndecan binding domain, which one can identify. And then one can, just like the same trick played in the previous slides, fuse it to a growth factor that I care about. The idea was that we anchor it to the cell surface by a non-signaling receptor, and the delay its endocytosis on the or the endocytosis of the receptor tyrosine kinase. Meaning, could we get prolonged signaling, which we called tonic signaling? We're not the only ones to use that word. So, just to illustrate the point of tonic signaling, this is with PDGF looking at its upstream signaling, meaning is the receptor phosphorylated? So, wild type phosphorylated very quickly, but then the amount of phosphorylated receptor goes down because it was destroyed. So, meaning signaling was quick. And if I look at downstream signaling, so AKT signaling, ERK1/2 signaling, phosphorylation also drops very quickly. But if we syndecan binding, that fusion protein, if we anchor it to the cell surface by a non-signaling receptor, we abrogate the peak, which I think had to do with the vascular permeability bit that I showed you, but we get a very prolonged signaling. So, what would have been gone in like 30 minutes max is here still going strong at 360 minutes, and we got tired and walked away. That's with many growth factors too. And does that result in an outcome? So again, back to the DBDB mouse. So here, putting these molecules in a fibrin matrix through an enzymatic cross-linking chemistry that we cooked up a long, long time ago. This is a substrate for factor 13A and fibrin. But here with the syndecan binding variant, so you can see really good healing. With the variant that's not syndecan binding at the same micromolar dose, yeah, not so much. And with the wild type molecule not anchored in the fibrin, even less so. And if you look at the amount of angiogenesis, sorry, you're looking at CD31 staining, you see this is actually double staining for either a pericyte or smooth muscle cell and Desmond, endothelial cell and CD31. It's a lot more interesting. So that concept of tonic signaling it really influences many of the thoughts that we have in our laboratory on the use of protein signals that bind to cell surface receptors, whether they're growth factors, whether they're immunological cytokines. So we've done a fair bit in the context of chronic wounds, and I'm going to I showed you some of that already, but with other molecules as immobilization concepts as well. But I'd like to go for a moment to, sorry, I put the wrong slide. I'd like to go for a moment to asking about cancer immunotherapy. So in our immunoengineering life, we think about immunity in the context of either making tolerance, like in the case of autoimmunity or inflammation or wound healing, we think of as making tolerance, I'll cover that in a minute, or to breaking tolerance. How do we interrupt a tolerogenic cycle? Meaning there would have been tolerance in the tumor, and I want to destroy that tolerance to allow rejection of the tumor. So here, tonic signaling, matrix immobilization. So that's what I'd like to go to. So how would we, and Gordana mentioned some of the concepts, how would we immobilize a cytokine that I care about? I don't care what it is, just a cytokine or banana flavoring or whatever. How would I immobilize that in the tumor microenvironment? And an approach is to take such a matrix binding protein or domain like PLGF2, that domain, use it to a cytokine or an antibody and inject that in the tumor. We did that quite some years ago with intratumoral injection of, for example, as I recall, an IL-2 variant that had a PLGF2 domain on it, as well as immunological antibodies like an anti-CD40. I'd like not to talk about that. So you could say, well, why not just put that PLGF2 bit in the blood? And the problem is it binds to fibrinogen also. So meaning it all gets very complex because there's, a mg per mil of fibrinogen in the blood. So we turn to other proteins that circulate already, and asking, could we utilize circulating proteins that bind to a matrix component and make fusion proteins? So I'd like to go, since tissue talks, we hear about macrophages quite a lot. I like to think about macrophages and how to modulate macrophage phenotype and DC phenotype in the tumor microenvironment. The earlier work was with IL-2, it was with IL-12, IL-7. Earlier, IL-7 was like last year, so it's not that old. But I'd like to talk about macrophages and dendritic cells, which are really interesting to bias, either pro-inflammatory or more pro-tolerogenic, pro-regenerative. So what is our targeting strategy? And I'm going to correct the word targeting in a minute. So we said, well, gosh, what proteins circulate already that bind to collagen? Or we chose collagen as a target. Collagens, because collagens are in every tumor. Collagen's solid tumor as a target. So von Willebrand factor, you may or may not know the protein, but it's a very large, complicated protein with many functions. One of which is to bind to denuded vessels to the subendothelium, and then it presents a ligand for an adhesion receptor on blood platelets. And it's the very beginning of thrombosis, which harkened me back to my graduate student years. So it turns out to have a 20 kilodalton domain on it called the A3 domain that binds with about 10 nanomolar KD to collagen 1 and collagen 3. And there's lots of collagen in the tumor microenvironment. So we thought we could fuse that domain to molecules of interest and get retention in the tumor microenvironment, not targeting, but retention. And at the beginning of this work, several people said, well, Jeff, that's stupid. But there's collagen everywhere in the body. So if you inject this stuff in the blood, and that was our intention, I didn't say that before. If you inject that stuff in the blood, there's collagen everywhere. It's going to go everywhere. There's going to be no selectivity at all. But we then said, well, no, in the tumor microenvironment, the vasculature is permeable. And I'll come back to that in a second. That's the way every protein drug administers in blood gets into a tumor. PD-1, PD-L1, every protein drug gets in the tumor that way through the leakiness of the vasculature. Now, there's argument about just how leaky it is, whether it's leaky to 10 nanometer particles or 50 nanometer particles or 100 nanometer particles. And I didn't want to enter that argument. We just said it's permeable to molecules. So are certain molecular drugs. They're protein, soluble protein. No one would argue that it's permeable to that. Whereas in the healthy tissue, it's not permeable. So there's like an and signal, decision between where there's vascular permeability, then there's access to matrix, bind to matrix, and be retained there. So did it target the matrix? Target the tumor? Not really. It just goes in the tumor and it hangs out there for a long time. It's the permeability that allows the targeting. So I'd like to go away from macrophages to a moment to just show you biodistribution in a really, really challenging model with an antibody. So a larger molecule, you'd say vascular permeability, a larger molecule. This is an anti-GITR. That's a fusion protein that my colleague in this work, Jun Ishihara, made in his lab at Imperial. And then Oscar Osuka, who's an immunotherapy guy at the University of Alabama, Birmingham, tested in a glioblastoma model, an orthotopic glioblastoma model. I'm going to skip over and not talk about this as a therapeutic biology. I just want to show you the targeting. So this is in the tail vein, injected in a tail vein in an orthotopic GBM model. Unmodified antibody in white, modified antibody, collagen binding domain, CBD in gray. So you see a lot of it retained in the tumor after injection in the tail vein. And now if I look at the adjacent normal brain, so there, it's been not very much at all because the vasculature was intact. So I have had people say, no, Jeff, no, we should use some antigen to target that's unique to the matrix of the tumor and not every place else, so that we can get better quality targeting, or better quality distribution is the word I should use. But then I argue back and I say, well, gosh, really? I mean, you're going to do better than, what is that, a 20-fold ratio? I don't think anything is going to do better than that. So I don't need specificity. I just need a detail of physiology. So now that's the biodistribution stuff. I want to go back to macrophages. So and DCs. So it could be quite interesting to think about GM-CSF and ask, can we use GM-CSF to modulate myeloid phenotypes? And so thus we'll make a CBD GM-CSF, the fusion protein of it. We show that it has the same receptor ligation, the same EC50 and signaling, all that stuff. And I just want to focus on the immunology for a moment. But if I look at this is in orthotopic melanoma, and so you can see the treatment points at the arrows, at the black arrowheads. But so if I look at survival, just to go quickly, so at collagen binding GM-CSF, and we had done earlier work with a very low dose of collagen binding IL-2. So I get a nice survival benefit there in a model that, for example, checkpoint blockade doesn't touch at all. If I use the wild type cytokines of the same molar ratio, so meaning the micrograms is on the cytokine basis, so that's in green. There's a little bit of joy there, but not nearly as much as when I immobilize them in the matrix. So this turns out to have a really interesting macrophage and DC story, meaning myeloid story. This is a very difficult model. It's a genetically engineered mouse model of an inducible tumor, inducible melanoma that with the tamoxifen that expresses the mutation of BRAF, a P10 deletion, and then stable expression of beta catenin. A model developed by Tom Gajewski at the University of Chicago. And its tumor mutational burden is very low, so it's difficult to address by immunotherapy. Um, and it's quite cold, meaning it's not inflamed at all. So it's very resistant to checkpoint blockade. You can see that so this is growth. I don't have the controls. The mice are quite precious. I mean, the no treatment controls. But PD-1 does nothing in everybody's hands. So that's our control. And then PD-1 with monotherapy of this collagen binding IL-12, meaning an immune agonist. So here, it's also not very encouraging. It does a little bit, and you can see the treatment points. But when we bias myeloid, and I'm going to come to the details, myeloid phenotype with GM-CSF in combination with IL-12 and PD-1, you get these progression-free survivors. And this is a really, really, really difficult model. And so, here we stop treatment at, what is that, 55 days. There's some animals still growing strong at 100 days. That's very challenging to achieve. How did we do that? It turns out to be more complicated than I expected. So here we're looking at macrophages and their inflammatory state of macrophages in the tumor microenvironment. So red is IL-12 therapy alone, green is GM-CSF therapy alone, always CBD, and purple is the combination. So we got a really good response with purple, but not so much with green, or red. So let's look at the differences here. So the inflammatory state, well, not, and this is the B16F10, not so different actually. And if I look at like macrophage numbers, not so different actually, meaning plus or minus GM-CSF. Meaning the combination. So GM-CSF itself induces a lot of macrophage infiltration. That does it. But then am I able to activate those cells? Not so much. So I'm not, as GM-CSF is very useful, maybe not therapeutically, but very useful at inducing tumor infiltration. If I look at, for example, the ratio of M1 to M2, if you use those words, sorry if they're too crude for you. Yeah, so you do indeed bias them toward M1, but you add the IL-12 in, you don't change things very much. So none of this really explains why we got such a better therapeutic response with purple than with green. Let's look a little bit deeper. So here, and the T cell response, and then we'll look deeper. So when we look at the T cell response in those tumors, there's really big differences. Remember, on the myeloid side that we showed you, there wasn't much anything at all. But here, like GM-CSF therapy, I get very low levels of CD8 T cells in tumor microenvironment. Or if I look at the ratio of CD8 T cells to Tregs, very, very low, meaning that's not good, that's bad. So whereas very different responses. So how does the previous slide, where there wasn't much difference between purple and green, how does that jibe with this slide, where there's a lot of difference in purple and green? So if you look at the cytokine milieu in the tumor microenvironment, there between purple and green. Really, really big differences. So meaning pro-inflammatory cytokines that have a strong and chemokines have a strong anti-tumor benefit. They're quite highly upregulated. Huh. How did that work? So that correlates with therapy. So thinking about this is now I'm going to focus on DCs for the moment, but thinking about biasing in under inflammatory conditions, I thought this result was very interesting. I mean, bad things happened. Um, uh, sorry, hope you can see the slides again. So if I lost my train of thought. So yeah, now focusing on DCs, differences between green and purple, as it were. So let's expose the animals to GM-CSF in vivo, and then let's isolate dendritic cells from those animals, and let's then stimulate them with IL-12. So interestingly enough, stimulation with IL-12 in the pink, when it's been pre-exposed to GM-CSF, upregulates the number of receptors of both subunits of the IL-12 receptor. So meaning that GM-CSF made the dendritic cell more sensitive to IL-12 signaling. I thought that was really interesting. And this is then that signal. So does that receptor signal? So it also more potent signaling. And then here's where the magic came in then, or whatever the good news, the joy is if I look at what happens when I pre-exposed to DCs, I didn't. Sung Kong and Eslen Mansurov did. Exposed to GM-CSF, I get a lot more pro-inflammatory cytokines produced by those DCs, interferon gamma, TNF alpha, and so on. So I thought it was very interesting here, biasing myeloid phenotype by one agent, and that increases their sensitivity to a second agent, IL-12, and gets a very interesting, strong anti-tumor immunotherapeutic response. I found that result really interesting. If you want to read the details, the paper came out a few months ago in GITC GITC. Uh, so we're interested in this approach of matrix immobilization, long-term effect, tonic signaling with a number of T cell targeting molecules, of BC and macrophage targeting molecules, and so forth. I just want to close that story with one last slide of, is there a difference in memory that's induced by those DCs? So here we do the therapy that I just mentioned, and we take the survivors, the complete responders, meaning they've gone on long after therapy has stopped. They're complete responders, and then we re-challenge them with the melanoma, the same melanoma, someplace else. And there's no more therapy. So we're just asking, does the mere presence of those melanoma antigens induce an immune response so as to kill them? Again, no additional therapy. So it's a memory recall cytotoxicity assay in vivo. And you can see that when we had past tense treated them with the combo with GM-CSF, all the animals resist the re-challenge. Whereas it's not so exciting. Well, it's exciting, but it's still not as impressive with the IL-12 alone. And if you look at antigen-specific T cell response, so these are cytotoxic T lymphocytes. They're specific for melanocytes, not necessarily tumor, but melanocytes. So here, if I look at the time course of expansion of those cells in the blood, it's with the monotherapy of IL-12, not so much. But when we biased the myeloid phenotype to produce all those pro-inflammatory cytokines in the original therapy, you get this really nice, very pleasing memory recall with no additional therapy at all, such that like 1% of cells are in the blood or antigen specific. So that's with those slides. Uh, what I try to show as an example of how we could use the idea of tonic signaling and localization to a microenvironment so as to get an interesting therapeutic response in the context of cancer. I'd like to go to regeneration now. So I'm going to go away from breaking tolerance, back to making tolerance. And I'd like to stay with myeloid cells. And others have led the way in biasing macrophages toward M2 phenotype, if you use those words again. And we were thinking about wound healing and how we could do this in a practical way with topical administration, in this case with interleukin 4. So well-known cytokine, difficult to handle systemically, but locally, I'm not concerned about its toxicity in Th2 immunity at all, but in terms of biasing macrophages. Well understood biology. The story gets boring to you maybe, but not to me. So Jun Ishihara and Aslan Mansurov and Akko Ishihara led this work with Abby Lauterbach. So here, DB mouse again, and we're going to do two kind of experiments. We're either going to look at healing outcome after 10 days of therapy, single administration, or we're going to sack the wound, as it were, and do flow cytometry and ask what's going on. So if we look at this, this now collagen binding, so as to mobilize in the microenvironment and get a tonic signaling effect versus a non-tonic signaling effect with just wild type IL-12, and the hyaluronic acid just keeps it from dribbling off. So there is actually there's a strong statistical healing benefit. So here is these animal experiments are very noisy, but 002, 003 there. So we do get a strong healing benefit, whereas with the wild type one, 0.3, we do not get a benefit in healing the response. But if it's more interesting to look at that's interesting to me because it's the rubber meets the road. But if we look at the flow cytometric readout of immunity, arginase 1 being in the macrophages, being an indicator of M2 macrophages, the one that I want, the pro-regenerative ones. So one sees a very strong elevation of those pro-regenerative macrophages under the influence of the therapeutic. And then if we look at cytokines in that wound, at that 3-day sac point by Legend-Plex analysis, so interferon gamma, that's a bad actor, it's downregulated. Interferon alpha, downregulated. GM-CSF is known to be associated with a better healing response, and it's upregulated. So GM-CSF is quite a dose response sensitive molecule, but upregulated, and that's known to be beneficial. So Abby led this work with Kirsten Refvik, published in NJP Regenerative Medicine. And so if you look at receptors, interesting question. So like we treat it with interleukin 4. And interleukin 4, interestingly enough, is known to downregulate expression of its own receptor. So we use this as a readout to say, indeed, IL-4 was signaling there because we saw its receptor downregulated as it should. And like I already showed you, arginase 1 upregulated. If you look at vasculature, so proliferating cells here with Ki-67, but CD31 endothelial marker, there's a lot more angiogenesis going on by biasing, not with an angiogenic growth factor, but by biasing macrophages, which are producing then several angiogenic factors. So yeah, a nice, interesting result. So to us. So I'd like to switch to another molecule, and I'll wind up and leave at least 10 minutes for discussion, but also focused on myeloid bias. But I'd like not to think about proteins at all now. I want to think about microbial metabolites, and we're having some fun with these microbial metabolites. Microbial metabolites like butyrate, and I'll just focus on butyric acid, produced in the colon, in the distal gut by fermentation of dietary fiber. Ri Wang, who's now at Wuxi, and Shiji Kao, who now has his own lab in PharmSci at University of Washington, led this work. In the gut, you produce about 7 grams of butyrate each day by fermentation, but almost none of that leaves the gut, and I'm going to come to that in a minute. It's all consumed in the gut and immunologically active in the gut. It's a major energy source for enterocytes, but it's known by action on a couple of GPCRs, and we're focused on its HDAC inhibition capacity to drive expansion of Tregs through biasing of dendritic cell phenotype. So it's as a drug, it's very difficult. You can buy it from Amazon. That's actually a screenshot from an Amazon webpage. It stinks. The stuff it sells tastes and smells like vomit. So it's a difficult molecule to deal with, and you have to use large doses because ZC50 is very high. So Shiji and Ru Yi cooked up for the goal of delivering butyrate to the distal gut in the context of dysbiosis, when there wouldn't be very much butyrate produced, cooked up polymeric micelles. And I'm going to come back to tissues in a minute, where the butyrate conjugation through ester linkage drives micellization of an otherwise water-soluble polymer. And I'm going to going a tad slower than I had hoped. But we then were able to show restoration of barrier function in dysbiosis. And this paper was on the context of peanut allergy, making animals less sensitized animals less sensitive to peanut protein that would be leaking out of the gut in the case of a dysbiosis. And so anaphylaxis in the mouse is measured by either death or a reduction in core body temperature. And you can see that treatment with these butyrate-containing micelles really normalized a strongly anaphylactic response. I want to skip a few slides. I hate when people do that. And to go to tissues, other tissues. So I mentioned 7 minutes left. I mentioned that almost none of that 7 grams a day produced in your colon gets into the system. It's almost all consumed in the gut, in the colon. So we asked, gosh, we saw some really immunological differences that I didn't show you in those animals in the gut. What would happen if butyrate were out in the system, systemically? So, gosh, how could we get butyrate out of the gut into the system? Well, amino acids get transported out of the gut pretty well. That's part of their role. And through transporters. Those transporters are not very specific molecularly. They're very sloppy. So what if we were just to do something simplest, one of the simplest molecules we've ever made? What if we just took butyrate, esterified it to a hydroxylated amino acid like serine? Would it get transported by amino acid transporters on the lumen beyond into the vasculature? And then moreover, those amino acid transporters are present on the cell membrane, so they could get into the cell where they could then inhibit an HDAC. Butyrate is a strong HDAC inhibitor. And so if we you can forget about the colon for a moment. One of the problems is that butyrate smells and tastes like vomit. I mentioned that, and you need a lot of it. But this serine conjugation totally blocks that smell and taste. And I can tell you by personal experience, that's even without the mint and the lemon, it doesn't taste and smell. So what does it do when it's in the system? So first of all, does it get in the system? So here, if you measure the amount of butyrate in these are normal biotic animals. So just amount of butyrate in the plasma in a normal mouse, when we treat it with a serine conjugate of butyrate, or when we just administer sodium butyrate like you'd buy from Amazon. So it's everywhere. And interestingly enough, those same transporters are on the blood-brain barrier. So we get it also amount in the brain normally, spinal cord normally, but the serine butyrate gets all the way there into the across the blood-brain barrier into the CNS. Might it be good for something? So let's go to inflammation and myeloid cells, and then I'll wrap up. So here, Erica Budina and Shiji Kao. So there's Erica's name, Erica Budina, who's fortunately joined me here at New York University, where I am now, and Shiji Kao. We're working in a neuroinflammation model, the EAE model, which somehow reflects multiple sclerosis or some features of multiple sclerosis. There's a myeloid imbalance that leads to priming of both Th1 cells and Th17 cells and leads to disease. I'm going to skip some of the details here, but if I look at the disease score, so these animals, this is just oral administration. So we gavaged it in this case, but oral administration. So they're administered, and I look at development of paralysis in the non-treated animals. So an animal with a score of 3 really drags itself about by its forelimbs, and its hind limbs are more or less paralyzed. An animal with a score of 1 does not flick its tail properly. And so, there's a really non-linearity in the score system. So just this very simple molecule butyrate reduces pathology. And then also, if you look at the CD45, all immune cells, hematopoietic cells, migrating to the spinal cord, much, much less. There's a histological benefit. There's a clinical benefit, but and more histological benefit. But I wanted to come to myeloid cells and T cells, and then I'll wrap. So here, if we're looking at the CD11B positive myeloid cells, using MHC2 expression as an indicator of their activation state. Indeed, the myeloid cells, and this is in the spinal cord, so are much less activated. There are fewer of them and much less activated. And so also, if I look at CD11C positive DCs, less activated. And if I look at the number of Th17 T cells in the spinal cord, many, many fewer. And if I look at the amount of pro-inflammatory cytokines in the spinal cord, much less. So this paper has a lot more immunology in it than this that I'm not going to show you, so as to not take too long. I like movies, these are animals that are untreated, just so you can get an idea what a score of 3 looks like. They lose a lot of weight because they don't feed well, and these are animals simply with the serine butyrate, so which look relatively normal. As I said, when a blinded observer views them, they observe a defect in tail flick. So, do these myeloid cells drive regulation? And I'm going to close on that. So here we're looking at meaning regulatory T cells. So these are in the CNS draining lymph nodes, the one that drains the brain, the one that drains the spinal cord. But if we look at the number of CTLA4 positive cells, a very important feature of regulation, elevated, or the number of FOXP3 CD25 Tregs, elevated. And really interestingly, to me, if we look at CTLA4 on those FOXP3 CD25s, also regulated. So upright. So this is an indicator of quality, not quantity of the T cells that's here, but this is quality of the T cells. So they're more potent in regulation. And so a close, like, can we induce antigen-specific Tregs without administering any antigen at all in this therapy? Because the disease state is producing the antigen. I don't have to do it exogenously, it's endogenous. So this is the antigen in that model, MOG. So if I look at just T cells that recognize MOG, it's quite suppressed. And if I look at Th17 T cells, quite suppressed. And if I look at Tregs that recognize that antigen, quite elevated. And back to where I find joy, if we look at the quality of those Tregs, really, really elevated. So we're driving this forward in a number of contexts where macrophages and DCs play an important role. Taryn Beckman and Lisa Volpatti, who's now running her own lab at Northwestern, have some really exciting data that's in revision in Nature of Biomedical Engineering. We'll see if we survive that gauntlet. So with that, this is the team. It's still in Chicago, right across the street from our building. So Erica Benina is not in the picture. She's joined at NYU. So Melody Swartz was involved in many of the immunotherapy, the cancer immunotherapy, protein engineering, and the CBD stuff was really cooked up initially by, Jun Ishihara is running his own lab at Imperial, and Aslan Mansurov, who has a fantastic job at Gilead. Thanks very much. I'll leave 10 minutes for questions.
Thank you so much, Jeff. So great talk, great photo, great that you left some time for questions. So let me just super quickly introduce the next week's speaker. So this will be Tejal Desai from Brown University, and you know that she is one of the big names in the material design and utilization for control of biological processes. So let's go to the questions. I'll start from the question posted by Dilip Thomas. Thank you for the wonderful talk. Typically, antibodies target towards cell surface protein that are targeted towards cell surface proteins have a slower dissociation kinetics. So the question is, how long does the tethered cytokine or growth factor last under in vivo conditions?
Yeah, that's a very good question. We've characterized that mostly in the tumor microenvironment, and it depends on the details of the proteolytic environment of the tumor. When the matrix is degraded, you lose it. And so there's a degradation component, so there's a K off component. But when we look by ELISA, we see different tumors like between 5 and 7 days, the action is more or less gone. The most meaningful readout, I thought, was in looking at IL-12. I didn't show any of those slides, but IL-12 leads to the expression of interferon gamma. That's sort of the most proximal response. And so what we did was we sacked animals and measured in their B16F10 tumor over time interferon gamma expression. So meaning that's the pharmacodynamic readout, and that lasted at least 5 days, and we didn't measure any longer after that. But it really hadn't decreased at all over that 5 day period. So I think that sort of time scale, not months, not weeks, but maybe a week.
Okay, so maybe a little bit of a general question. I mean, it is very well known that the interactions between immune system and cancer cells are very much dependent. You know, they change like for the same patient at different times between the patients, etc. So what is the situation with immune regulation of the environment that you discussed in the context of inflammation and the context of regeneration? Are these
Yeah,
findings?
that's really interesting question too. And so maybe I'll first answer part that
Sure.
wasn't asked about in the cancer microenvironment. So for example, some tumors are quite exhausted. They may be very inflamed, but quite exhausted. And the 4T1 model of triple negative breast cancer is one such model. And their IL-12 doesn't touch it at all. Whereas it totally got complete responses in B16 or EMT6, another TNBC. But when we added there an anti-exhaustion cytokine, IL-7, then we rescued that effect. We got there a very strong therapeutic response. So I think different microenvironments, the myeloid cell might be troubled, the T cell might be troubled, for example, by exhaustion. In inflammatory sites, meaning not a tumor, but like an inflamed bowel. So we've done less work on immune characterization there with these cytokines on a CBD. Rather, we've done more. We've several papers using a lymph node targeting approach, where we administer subcutaneously, and then a bias immune education, the lymph node that changes the disease site. But in the actual inflamed microenvironment, we've done less work.
Okay, but then it looks from what you say and from the literature, it's really like these results are more generalizable,
I
like
believe so. Yeah.
yeah,
Yeah.
rather than the other.
Yeah, so with the cytokines that target lymph nodes, so they're the EAE models to go to, but so is our two different rheumatoid arthritis models. We get very similar results in both of them, and we have some interesting pulmonary results as well. So yeah, I think it's well generalizable, but there may be situations where, for example, T cells are exhausted, or where we could generate a Treg that's contravened by another factor.
And using the models that you developed, is it also possible to look into some of the biological factors like age or presence of a specific disease, etc., on the progression of, say, inflammatory and regenerative processes?
I think it is actually. We've got a little effort going on in the lab, on inflammaging, so looking at these responses, including vaccinal responses, but as age goes on, and so we're quite interested in setting. Haven't started yet, but setting up experiments, where we start treating animals when they're quite aged, and then asking whether we can modulate the inflammatory state in those latter weeks, in those cases, or a few months of life of those mice. So I'm really interested in asking, especially with an oral molecule like butyric acid or the systemically available butyrates, like can I just bias immunity toward tolerance? With something I drink, and whether we could change the inflammaging processes associated with that. I should I add though that you'd say, well, am I immune suppressing? Is this like taking a steroid, a cyclosporin or Humira? But in these studies, we usually set up experiment where we vaccinate an animal with a typical adjuvant. Like MPLA plus alum, and ask, do we abrogate the vaccinal response? And so, like in that butyrate study that published in Nature BME with Shiji and Erica, so there we vaccinated and we looked at humoral responses and cellular responses, and did not see any statistical abrogation in either case. So I think in the face of like a hardcore inflammatory signal, like a vaccine TLR agonist, we did not suppress immunity. We haven't done though like flu challenge or something like that, asking whether we blocks the system or weakens make the animal more susceptible.
And maybe just to ask you, like, about you describe different states of the cells that are at least in part induced by the environment. How much dynamics versus stability is there? So when the cell assumes a state, I mean, how much does it take to perturb it? And how much reversibility, if you like, is present in this?
Yeah, I can only that's a very general question. As you would well agree, it depends on the details of the state and the perturbation. But in the cancer context, it's often if we stop therapy in the absence of checkpoint blockade, we don't necessarily get a whole lot of complete responses. It depends on the immune state of the tumor. But in the presence of checkpoint blockade, we get great memory, like I showed with that GM-CSF study, meaning it's quite robust and stable. But in the tolerance side, I think in our antigen-specific inverse vaccine approach, the overall goal has to be, maybe I overstate, generation of regulatory T cells, which have memory. So there, with some of our studies, like in an allergic asthma paper that's in press now, in Science Transition Medicine from Emiliano Gomez Medellin. There we could challenge and challenge and challenges for like 150 days without any therapy, and maintain a tolerogenic response because there were Tregs. And when he deleted Tregs after therapy, you completely lose that effect. So that's why I think I'm excited when any of these approaches, like the Syrian butyrate or cytokines, when we generate Tregs, then I feel really happy because I think about longevity, or rather durability.
Yeah, maybe just one more question because we'll need to let you go soon. And then for the remaining ones, we would like to give you contact information and maybe, respond offline. So Jeff
Yeah.
Zosserman is referring to the work done by Hoffman and David Baltimore, who showed that the kinetics of NF-kappa beta activation can differentially regulate targets. It looks like you have similar responses. Have you identified how the signaling dynamics is causing this? Like what is the
Well, I wouldn't
relationship
say in,
signaling?
in as much detail as in that study. What we do though is, like with the immunological cytokines, we do look at phosphorylation of the relevant stats, and even some that are less relevant, to try to understand who is being phosphorylated and how long they're being phosphorylated, they're staying phosphorylated. But that's a deep question within what happens after stat phosphorylation downstream of that. And so I don't think we've characterized that super well. But what we are trying to do though is engineer some of these molecules to ask, how could I trigger phosphorylation of different stats that I wouldn't necessarily have seen, so as to modulate and bias the phosphorylation event. But in terms of the dynamics, like you know um, 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 Savas Tai has looked at like noise in NF kappa B signaling, and seeing even that like noise makes a difference in the cellular outcome and signaling outcome in things that I don't well understand. But there got to be 1,000 details there, and we probably just looked at one.
Jeff, thank you again. This was really amazing. Thank you so much.
Yeah,
And
thank you so much for the opportunity to join.
sure, sure. And thanks everyone for very nice questions. Okay, see you next week.